while the projector's coming up here. What I want to do today is start by telling you about the test that you're going to have next week, the practical. And then I have a few things to say about skin. Last week I had said I would get a chance to do that, and I pushed the due date on the skin stuff back. I realized just this morning I never did change the due date. And so that's been done today. The skin stuff is officially due next week. If you've already worked on it, that's fine. Most of it really you can get out of the reading. I just want to say a few things that are more about how the chapter fits into the course. And then we're going to move into the skeleton. And I want to talk about a few things about the skeleton chapter, mostly about bone growth. And then we'll spend the rest of the day looking at the skeletons. My hope is that we'll do everything up to the bone growth stuff in the first half, and then in the second half, we'll look at the skeletons. But my plans don't always work out. So we'll see what happens. So to begin with, I want to talk about the practical. There's a folder specifically that says Lab Practical 1, and I want to point you to this. If you've taken a laboratory science course before, biology course, you might have had a practical. That's what we call a laboratory exam. It's based on the model sort of from a more classic biology course, which the modern equivalent is like medical school anatomy. So in a medical school anatomy course, the students spend the course dissecting a cadaver, and from that studying the anatomy up close and personal. And they're tested on what they know in what's called a practical exam. And the word practical here is sort of referring to it as being the practical information that they're learning. They're being tested on the dissection. So in the most classic sense of this, the students that were working on a particular dissection would meet with the instructor, and the instructor would basically sit there and have them name all of the different things that the instructor points out. Translating that to a course like this, we don't do dissections per se, but we have models representing dissected material, such as this vertebral column model right here. So what a lot of my colleagues do, and it's sort of the more common way of testing on lab material, is to have the room set up with a bunch of models and specimens and microscopes, that sort of thing. You'd get a regular stapled sheaf of papers handed out to you at the beginning, and you'd go around to go around the room from station to station and answer questions that are about that thing. I don't do the testing quite that way for a couple of reasons. One, setting up that kind of test is a little bit unwieldy. It's kind of hard to come up with all the questions and set up things semester after semester, especially as This course has gotten to be more popular, and we don't have a whole lot of time between labs. It takes about an hour to set up that kind of test, but sometimes we only have 10 or 15 minutes between labs. So it's not the most efficient way to set up a test. So several years ago, I switched to a different sort of format, and that's the way that it works here. The practical has been sort of redefined, thinking of practical meaning being tested on real-world practical information. And in that, the test is based on case studies. And we've already looked at one case study. And when we did, we looked at that cystic fibrosis case, which is actually what I have here. That actually came from this document here. It says practical 1.1 cases. Now, this is our first practical, so it's practical 1. And the 1.1 is just code for me to keep straight the A&P1 and A&P2 stuff. So if this were A&P2, it would be practical 1.2. So that's what that code there means. And then this is just a document that has 4 case studies laid out. The second one in the series is the cystic fibrosis case that we did in class. The first one is a bit more, it's a smaller case study, kind of a quick little one that's meant to address some of the introductory material from chapter 1 of the book, sort of where things are in the body cavities, that kind of stuff. And then the cystic fibrosis case, which we studied in depth, has to do with the molecules and cells and tissues. And then after that, we get into, there's 2 more cases down here, both of which are about broken bones. One's a motor vehicle accident. The other one's a woman with osteoporosis. So those 4 cases address pretty much all of the stuff that we've done so far this semester. So there's the first case. It's really about the introductory, where things are in the body. The second one, which is, introductory sort of chapter 1 stuff. The second one, that's kind of chapters 2, 3, and 4. And then cases 3 and 4, they get to chapters 6, 7, and 8. You'll notice that I skipped over chapter 5 there. That's the skin chapter. There's not a case specifically about the skin. And as I talk about the skin in a little while, I sort of treat it as an extension of tissues. So the tissues, to some extent, fit in with, skin fits in somewhat with the tissue stuff, which is in case 2. So the cases kind of cover everything for the so far in the semester, the first half of the semester. And the test is going to be directed at or following this information. Instead of the practical exam I described before, which was all about testing students' familiarity with the dissections they did, this is going to be a practical exam in how real-world examples relate to the material that we've covered. That's kind of where it comes from. The test has 2 components. There's an in-class portion, which you're going to be doing next week, and the online assignment, which is in that folder that I was just in, and I'll show you that in a little while. The in-class portion that you're doing next week is a 50 multiple choice question test, and it'll have these 4 cases on it. So you'll have a stapled set of pages that are the test. It'll start with a place for you to put your name, and then it'll say case 1, and it'll have exactly what's written right there. You don't have to memorize the cases, they'll be there for you, but you do need to be familiar with them when you come in. And then following the text of case 1, it'll have 12 or 13 maybe questions about that case. And I'll go into more detail about the questions in a second. And then after the last question on case 1, it'll have case 2, the exact case that is on there that we looked at before, followed by its 12 or 13 questions or whatever it is. And then case 3 with its questions and case 4 with its questions. Now I'm saying 12 or 13 questions. The number of questions within for each case varies just because 4 doesn't go into 50 evenly, so they're not going to all have the same number of questions. And so there's a little bit of variation, but there's about a dozen questions per case, roughly. Within that dozen questions for each case, they fall into what I like to think of as 3 kind of categories as to what the questions are asking about. They're all multiple choice questions. And the first type I describe as questions about the case. Now, when I described case studies, when I looked at the cystic fibrosis stuff to begin with, I told you that I expect students to look at cases and get information out of it by going through the terminology, the symptoms and signs, and the systems. We did that with case 2 and went into a little bit more detail about it. But for cases 1, 3, and 4, that's what I'd expect you to be able to do. You can look at case 1 and make sure you understand all the words that are there, and then pull out what symptoms are described and which body systems that has to do with. Now, case 1's a little special compared to the other ones. I need to give you a little bit more information, which is just that when you're looking at the systems, you're not, for any of these, trying to diagnose anything. That's not the point at this level. But with case 1, I want to give you the heads up when you're looking at the systems that are affected, since this case is really about the terminology that has to do with anatomy, and especially the body cavities and where different organs are located. What's going on in case 1 is he's injured an internal organ. From the symptoms, you should be able to figure out which organ has been affected. And just, you don't need to know so much about the organ per se or the organ system that comes from, but rather what body cavity it's located in. So that's really the focus of the case. To just give you a case about body cavities, it would be like a morgue case or something like that, cutting up a cadaver and looking inside of it. So just to give you a little bit more real world case, this is about a damaged organ, internal organ. When you're looking at it, at the systems part of making sure you understand what's going on, you should recognize what system's affected based on the symptoms that are there, and especially the anatomical clues will tell you which organ specifically is affected. And you just need to know where that's located. That's the primary thing about that. But anyways, so you'll get questions that are about the case based on the terminology, symptoms, and systems approach that I described to begin with. Now with case 2, since we spent a little bit more time talking about it, I'll expect you to know a little bit more about that going into it. But for cases 1, 3, and 4, that's essentially it. Now with cases 3 and 4, they're about broken bones. So when you're talking about what symptoms there are, the symptoms are this bone or that bone's broken. And then obviously the skeletal system will be the primary system that's affected, but also keep in mind how the skeleton relates to the organ systems that it protects, and that might be relevant to what's mentioned in the case. So it might be a question about what does a particular term mean in the case. It might be a question about a particular symptom. The cystic fibrosis case, she has coughing. So it might ask you, what do you call the substance that she's coughing up, or something like that, which would be kind of a symptom or terminology question there. And then could be questions about the systems. So it can get at what the case is saying and the underlying anatomy and physiology that has to do with it. The second type of question is what I consider a question that's not about the case, which might sound like I've kind of opened up to asking you anything. I have questions about the case and questions not about the case. And so logically, everything falls into one of those two groups. But what I really mean by questions not about the case is each case is about a particular part of the course. And I need a little latitude to ask you questions outside of the case, but still about that material. So case one, like I said, there's a particular organ that he's injured and it's located in a particular body cavity. But I might want to ask you a question about one of the other body cavities. So that's not going to be about the case, but it's still about the material that the case is from. Or in case two, the tissue that's affected is epithelial tissue that secretes mucus and that kind of stuff. But I might want to ask you a question about connective tissue or something like that, which would not have to do with the case because it's not a case about a connective tissue disease. Or with the cases 3 and 4, which are about broken bones, I might want to ask you about bones that aren't mentioned in the case. Or something like that. So those are questions that are not about the case. And then the third type of question is the more traditional station-based question. So I'll have stuff like this vertebral column model here, and I'll have pieces of tape on it pointing to things, and I'll ask you questions about what you're looking at. So this is not a question that's on the test, but let's say I'm showing you this, and I'm pointing at one of these pieces of rubber in between the intervertebral bones. I could ask you what type of tissue that piece of rubber represents. What would that be? It is in fact cartilage, but that's actually not going to be the correct answer. But you wouldn't know that because I'm asking you an open-ended question, but you'd actually get a multiple choice question on the test. So the answer choices there might be A, spongy bone, B, hyaline cartilage, C, fibrocartilage, or D, elastic cartilage. So I like 4 answer choices things, and there's 3 types of cartilage, so I had to put something else in there. So I went with spongy bone. Being the only choice in that it's bone, it should be pretty obvious that that's probably not correct because all the other choices are about cartilage, and you should recognize that that's going to be cartilage there. So it's a matter of which type of cartilage it is. So you know the types of cartilage, the names are all right there in front of you in the multiple choice question. Which type would we find here? Fibrocartilage. When I went through the cartilages, elastic cartilage is found in the external ear and the epiglottis. Fibrocartilage is found here in the intervertebral discs, in the pubic symphysis, which is here, and also in the menisci of the knees. Everything else lies in the cartilage. That was the way I sort of went through telling you which types of cartilage is. So yes, this here, anywhere that we're bearing body weight, we're going to have fibrocartilage. And this obviously is bearing body weight on the spine. But that's the kind of question I might ask. Now, that's not on the practical. That's a tissue question, which would go with question, case 2, and it's just not one of them. In fact, on case 2, there will be 2 stations, actually, there will be 2 stations for each case, and the 2 that are for case number 2 are both going to be microscopes that you're going to be looking at. So in that, for those questions, you need to be familiar with the tissues that we looked at through the microscope that I posted in that folder there. And I'll talk actually a little bit about that before we move on. So those are the three types of questions. Questions that are directly about the case. The case is on the piece of paper in front of you. You don't have to memorize anything, but you should come in prepared, having made sure you understand the terminology, the symptoms, and the systems for each case. There may be some questions that are not about the case directly, but about the material that the case is essentially over. Case 1 is about the anatomical terms and body cavities from chapter 1. Case 2 is about molecules, cells, and tissues, which is chapters 2, 3, and 4. Chapter 5 is about skin, and for this test that's fit in there, but you should remember that there is something about cystic fibrosis that relates to the skin, so be prepared for that. And then cases 3 and 4 are both about broken bones, so those are the skeleton chapters 6, 7, and 8. So I might ask you questions about that material, not specifically about the case. And then I'm going to ask you questions that are based on looking at things like this. Now, for the station type questions, I'm going to try to make them relevant to the case as much as I can, but there might be something that I really want to ask. And so if for tissues, I was going to ask you something about fibrocartilage. Doesn't have anything to do with the case, but this is a great example of fibrocartilage, a very easy question to ask. So I might have something like that, but I try to make them about the case as much as possible. Of the dozen or so questions, you can anticipate that 8 to 10 of them are going to be about the case itself, and then 2 to 4 are going to be about other things. Okay. I want to make this as much about the cases as possible, but I do need latitude to ask you about other things. You are responsible for everything, so I want to be able to ask you those questions. However, in studying, the case study approach does give you a little bit of a clue as to what to concentrate on. So like I said, case 1 is about anatomical terminology and body cavities. From the case, you should be able to figure out which internal organ's been damaged, and so you should know where it is. So be sure you know that body cavity the best, be able to describe the location of that organ using the terminology that's covered in chapter 1, and be able to relate it to neighboring organs around it. You know, is it anterior to, or posterior to, or superior to, or inferior to other structures around it, that kind of thing. So concentrate on that material first, and then you can try to go on and study other stuff too. Whether I describe this test on the first day or I describe it here the week before you're going to take the test, students never have enough time to study everything. If you're the kind of student that does study everything, you've been studying everything all along. If you're the type of person that doesn't have enough time to study everything, I told you about this on the first day, you wouldn't start studying for it until this week anyways. So the way I've constructed the test kind of plays to that a little bit. It helps direct you as to what you should study. Probably a student can effectively study in the week before a test, maybe 20% of the material that the test is really over. So if you study the specifics of the cases first, you'll maximize what you're studying. Instead of saying, well, I'm going to randomly study all the material in the course and hope I study the stuff that he asks questions about, Here I'm kind of telling you what I'm going to ask most of the questions about. So study this stuff first. The best way to kind of illustrate that is with the bone stuff. We haven't talked about the bones just yet, but there's 206 bones in the body. In those two cases, it probably describes a dozen or 2 dozen bones that have been broken, closer to a dozen. So if you're only going to study 20% of the skeleton in the time you have for the test, you're going to study about 40 bones. Okay, so be sure in those 40 bones you study the ones that are specifically mentioned in those cases. Know them best. Those are what most of the questions are going to be about. Okay. And it'll just help you be a little bit more efficient with your studying stuff. So for instance, in case 3 here, motor vehicle accident, the patient broke the tibia and fibula, a few ribs, and the scapula. Okay, so be sure you know those best. And then in case 4, all of the bones in case 3 are just right there in that section together. Case 4, they're scattered around a little bit more, but here she's broken a wrist. Here's ankle. Where is that? Here's part of her spinal column. Those sorts of things. So they're mentioned in the case. Pay most attention to those. And so between those, you're going to study, again, maybe about a dozen or so bones. You should have time to study more bones. Go on to other bones that I make a point of mentioning, because those are going to be the ones that are sort of the most important examples there. And when I get to the talking about the skeleton, I'll talk about a few bones in particular as examples, because they're kind of the go-to examples that we use a lot.
You mentioned something with the organs and knowing where they were essentially like each other. Do we just know what organs are in which cavity?
So that's just about case 1 that I was mentioning that. And so in the case, you should be able to figure out that a particular organ's been damaged. And so you should know that organ the best. Or you should know that organ in relation to the anatomical terminology the best.
Right.
So the case isn't about you knowing the various organs, but more about the cavities and that sort of thing. So for that, you should know which organ's been damaged, what cavity it's in, and how to describe its position anatomically in relation to other neighboring organs.
Okay.
And then
other random questions.
Right. Then it would be more about understand the anatomical terminology, more about the directions that are described in anterior, posterior, that kind of stuff, and the different cavities. Planes of section goes in with that too. So with direction, it's also the 3 ways that we can cut through the body to look inside of it. That goes with it too. But it's not about knowing all the organs.
Right.
I'm just saying for the case is about an internal organ that's been damaged. And letting you know at a time for the case that you pay attention to that.
Right.
But the material is really more about the anatomical terminology.
Okay. So we won't need to know for like the other random questions, like where other random organs are in comparison to each other, kind of, because I just don't know
Right,
that kind of stuff.
It won't be, if I do anything about comparing locations of organs, they should be very obvious organs like heart, probably the heart is,
Right.
or the brain, that kind of thing. It won't be something obscure. And it'll be more about, you should be familiar with cavities. Giving you the case studies to prepare for ahead of time, one gives us a little bit of framework for the test to be based on, but also helps direct your studying a little bit. So if you study the cases primarily, while they're not going to cover everything that we've studied so far, the majority of the questions I'm going to ask you are going to be about those things. So, for instance, with bones, you should know those bones. You should know where they're located. You should be able to recognize them. If you're presented with those bones, you should know what they connect to, what joints they make, that kind of thing. Know that stuff best. And I'll talk more about the bones when we get to that part of the class. So that's the in-class portion that you'll be doing next week. The test is designed to be completed in about an hour. And when I say something like that, the way that I come up with that is I take the test. I physically sit down, I read the test, I circle the answers, turn the pages, all that sort of thing. However long it takes me to do that, I can assume you should be able to complete the test about 3 times the length of that. It took me about 15 to 20 minutes to physically take the test. Of course, I know the answers, so I didn't have to spend a lot of time thinking about what's going on there. But just the physical act of taking the test took me about 15 to 20 minutes. And so that would suggest that you should be able to finish it in 45 to 60 minutes. So adding in the time that's required for you to think about and recall what you've learned about the course. That's sort of the rule of thumb there. The normal structure for a course like this, the laboratory component, we meet once a week for an hour and 50 minutes. And so traditionally, that's how much time I've made available to take the test. So even now that I don't teach that more traditional type of class, I still do that. If you need an hour and 50 minutes to complete this, you have that. We, of course, have 4 hours here. So my intention for next week is the first half of the morning will be set aside for the test. And then the second half will cover joints and the introduction to movement and muscles and stuff like that. Depending on exactly when everybody's finished, like I said, most people will probably finish in about an hour. We can get started on the second half earlier. So I won't say, okay, at 10 o'clock we'll come back. If everybody's done with the test at 9, we might as well get started on the other stuff sooner. And that just means we'll finish up that much sooner too. But more on the logistics of that next week when we actually know how long it's taking people to finish stuff. So that's what you're doing next week, taking that 50 multiple choice question test. Oh, I didn't mention anything. There's a link in here to an outline. I've never been one for outlines. When I was a student, I hated outlines. Writing assignments always started with, make an outline of the paper you're going to write, that kind of stuff. And I always did those just to satisfy that component of the assignment, but I never used outlines. I don't really care for outlines. So it's not the best outline. It's just something I put together several years ago now. The kind of outline, so to speak, what's covered in the first half of the semester. Especially since we don't spend as much time on material as I might have in earlier semesters. It's a good place where I put a little bit of information about everything. For this particular outline, actually, it starts off with sort of definitions of the anatomical terms from the first chapter. And then for the tissues, the microscope stuff that we did, it lists the different types of tissues, concentrating on epithelial and connective tissues. And that's sort of the first couple of pages of the outline. The rest of the outline, which is like 5 more pages, is actually just all of the different bones. And in the outline, I try to put one statement about each bone. It's just impossible for us in class to go through every bone in that much detail. So the outlines, in this case, is a good source for at least a little something about every different bone there. So it's a useful place to go. But saying that I don't really like outlines, I didn't as a student, I don't give them a whole lot of importance, I suppose. Some students will have the impression, and I don't quite understand how this ever came to be, but that an outline is like a legal document. If it's not in the outline, it's not in the test. It's not how I work. You're responsible for what we've covered this semester, and because of the fast rate of this semester, covering does not necessarily mean explicitly stated in class. The material that we've covered, chapters 1, 2, 3, 4 in Unit 1, and then in Unit 2, we'll have gotten through basically 4 chapters worth of material there too. But the outline gives you a sort of a clue as to, or not a clue, but a guide to what we've concentrated on in the course. Okay. So that's there too. Then the third thing that's listed there, that's another assignment. So that's the online component of the practical. It's an active assignment. You can open it up now if you want to. It was originally conceived as an assignment that you'd complete after the in-class portion. When I first started doing the test this way, you'd come in, you'd take the in-class portion, you'd hand that in, and I'd hand you physically a take-home set of questions. Those take-home questions ended up being incorporated into an assignment on Blackboard. And I used to hide that assignment until the day that people took their practical. I sometimes forget to do things online, so what I've done over the past few years is I've just made those assignments available from day one, so I don't have to remember to activate them at a certain time. So you are welcome to go in and look at that if you want to, but it's really kind of, like I said, conceived as being an assignment for after you do the test next week. There are 4 questions for each of the 4 cases, so 16 more questions here. They're more like the kind of questions in the Part 4 assignments that you've had so far. They're mostly open-ended. There might be some fill-in-the-blank type questions, that sort of thing. And there's you can't quite read it on the screen there, but that little list in the description there says questions 1 through 4 cover case 1, questions 5 through 8, et cetera, et cetera. So the first 4 questions about the first case, the second 4 questions about the second case, et cetera, et cetera. I just want to mention that because sometimes people aren't really paying attention to the question numbers and they start answering a question based on the wrong case. And on this particular one, that's the worst because cases 3 and 4 are both about broken bones. So if a question's asking about a bone and you go to the wrong case, you're going to be answering the question wrong. So pay attention to which case the questions are about. Now the in-class portion that you'll be doing next week, 50 multiple choice questions. The questions are each worth a point, so that's 50 points in that in-class test. Here, these are worth 2 points apiece for an additional 32 points worth of questions. 50 plus 32 adds up to 82. The test is actually worth 75 points. And while you can score more than 75 points, it's a 75-point assignment. And so if you do score 76 through 82 points, those extra points are extra credit within your lab score. Honestly, very few people get extra credit points here. More realistically, what it's about is giving everybody a little bit of breathing room. You'll be taking the in-class portion most likely before you complete that, and definitely before I grade any of the online portion. So you'll often think, and students think this way all the time, that your grade is, what you did in the in-class portion. But really, if you think about with the extra points, it's all one big assignment, one big test. Don't get too freaked out about what you do in class. You could answer 38 of the 50 questions correctly in class next week, and walk out if you're thinking that you're going to have what's essentially a C of the class in the test. But again, it's all one big test. So 38 points in class, and then online, you get 32 more points online. 38 plus 32 is 70. Okay. And 70 out of 75 is, I think, 92.5% or something like that. So the extra points that are built in there can actually make what seems like a dismal attempt on the in-class portion be okay in the end. Now, there are only 32 points worth of questions on the online practical, and the example I just gave there requires you to have gotten all 32 points. But actually, that's not impossible. Probably about half the students, I've never actually sat down to count this out, but it's always felt like about half the students in all my classes get 30 or 32 points in the online portion. The average is probably somewhere around a 26 or 27. It's not a normal distribution. It's not a bell-shaped curve or anything like that. It's actually skewed a little bit high. People tend to do pretty well on it. The average gets brought down by people that just don't get things or answer questions incorrectly because they're answering a case 3 question based on case 4 information or something like that, and people just kind of bomb out on things. So that brings the average down, but it's not uncommon for people to do really well on this. The questions are worth 2 points apiece. They're like part 4 questions, but part 4 questions are 6 points apiece. And so grading is different between the two types of questions. In part 4 assignments, if you don't quite get it exactly right, I can give you 5 out of 6 points. Take a point away just sort of to address the fact that you didn't quite get it. Sometimes people add a little bit too much information and get off on the wrong track and answer something like that. I can deal with that. 6-point question. But with 2 points, it really works out like this. If your answer is thoughtful, if you've answered the question as it's asked, and you obviously put a little research into what the question's about, you'll get 2 points. If your answer's in the right neighborhood, but not very thoughtful, or you over-answer the question, it's a question about tissue, and you start answering about all of the different types of tissue instead of just the tissue that the question's asking about, that sort of thing. Then you might get 1 point. If there's something correct in there, but you went too far with your answer, something like that, you might get 1 point. Or if you're just not quite on point with the answer. And if you're just completely off base, for instance, answering a case 3 question based on case 4 or something like that, you won't get any points. With that approach to grading, 2 people can answer the same question very differently and still get full credit on it. So it's not uncommon for people to do very well on the online portion. The idea behind the online portion is for you to go a little bit deeper into the cases. So for next week, I expect that you are going to be familiar with the terminology, the symptoms, and the systems with each case to answer the multiple choice questions you have. But then when you go to the online assignment, those questions are going to ask a bit more in-depth kind of things, and you're going to have to study the cases a little bit more. So it might ask you about how things are treated, what the eventual outcome of the condition might be, or, well, half of it's about broken bones, so it might be about how bones heal and grow and that kind of thing. So it can go a little bit more into depth. And so the questions will direct you to find more information from the case and about what the question's asking. It'll require you to go a little bit more in-depth and research and study of the material. And so what I'm looking for is that I can see people are doing that. exactly how you answer might not be as important. I mean, no, exactly what you answer might not be quite as important as how you answer. I see that you're doing the work to find that stuff. Some of this stuff that these questions are about is evolving, since our primary source for a lot of this is the internet. New things might have been discovered, or new articles might explain things a little bit differently. So sometimes people answer things and I've never seen that answer before. I take your answers, I look into them, and sometimes I learn new things and I find new ways of people answering stuff. There's a bit of latitude in some of those answers, and that's built in. So people do very well on this for the most part. If they take the time, they think about what the questions are asking, and they stick to answering just those questions. So that's there. That's essentially what the practical is about. Okay. So the in-class portion next week, 50 multiple choice questions about the 4 different cases. You should prepare for those by looking at the terminology, the symptoms, and the systems for each case. For the cystic fibrosis case, since we talked about that a little bit more, I anticipate that you know that a little bit better. But the other 3 cases, it's simply that. And then there's the online portion. Its due date will be for the week after. So let's see. Today's the 30th, so we'll be doing the test on the 7th, and the online portion will be due on the 14th. I think I have the dates right there, but something along those lines. Any questions about the practice? Yeah.
Is the online portion timed?
No, it's just like a part 4 assignment. So you can open it up, you can do a little work, save your answers, come back later, that kind of thing. If you want to, while you're preparing for next week's, if you want to look at that, it might give you a little bit of a clue as to what's going on in some of the cases. Because I'm asking more in-depth questions, so obviously I'm going a little bit more into the cases, and it might even help you understand what's going on a bit better. Other questions? Okay. So as you're preparing for things over the coming week, feel free to contact me with questions, text or email, or something like that. That's fine. Okay, so I was kind of hoping I wouldn't spend so much time talking about that, but I can never really do that in less than about 45 minutes. Where are we here? So I do want to say a few things about the skin. Actually what I'd kind of hoped to do, and it didn't quite pan out, was to prepare a sort of online presentation about this stuff. And so this is the presentation software I use. And I actually sat down and put this together and got it updated with stuff from this book. And what I can do within this presentation software is I can put sound files in, and so I can narrate the slides somewhat. And that was my intention to do. Didn't get to do that. Had some technical issues getting it to work the way I wanted to. But I do have this, and so what I want to do here is just talk about the skin briefly and point out what I want you to pay the most attention to. I'm going to leave you to get some of the information from the book in more detail, and I just want to talk about what I think is most important. Would somebody mind turning off on the lights over there? So, now, the way the course is set up, we start off in the first unit of the book, chapters 1 through 4. We're going through the foundational material. Okay, how do we use language to describe anatomy, the chemistry, the biology, and the tissues that help us understand anatomy and physiology. And then once we're finished with that Unit 1 material, we move into looking at organ systems over and over. So the first organ system we deal with is the skin, the integumentary system. And then we go on to the skeletal system, and then the muscular system, and the nervous system, and then the endocrine system, the cardiovascular system, respiratory, digestive, urinary, reproductive. We just keep going over different organ systems throughout the whole course. The skin as an organ system isn't that exciting to me. There are some aspects of it that can be pretty involved. It's a huge organ system. It's vital. If you didn't have your skin, one, you'd be really disgusting to look at, and two, you probably wouldn't survive it very long. But partly, I think because it's so ubiquitous and everybody has skin, it's there, we deal with it. I've always felt that it's not the most exciting organ system. I prefer things like the heart and, how do we regulate blood pressure and the electrical activity of the nervous system, that kind of thing. Skin just doesn't really have a lot to it. So I prefer to use this chapter as kind of a transition as we're going from that foundational material into actual organ systems. It's a great place to sort of start to think about how that fits together. And what I want to stress with the skin is to look at the tissues that are involved. And there are all the different types of tissues represented in every organ system. There's going to be epithelium, connective, muscle, and nervous tissue in every organ system that we're considering here. And so with the skin, we can look at which particular tissues are found there and how they relate to the function of the skin. So this is just a quick rundown of where we find the different types of tissue in the skin. The epithelial tissue that's most prominent in the skin is what we call the epidermis. And there's a stratified squamous epithelium that is the outer covering to your whole body. It's actually not the only epithelial tissue in the skin, but it's the primary one. And it makes up all of the layer of the skin that we call the epidermis. The rest of the skin is what we call the dermis, which is primarily connective tissue. But then within the dermis, within the connective tissue of the dermis, we also have other structures which are made up of other types of tissue. Muscle in the skin is a little bit of a stretch, no pun intended. There's not a whole lot of muscle there, but there is muscle. And the muscle tissue that's really part of the skin, most primarily, are the muscles that attach to hair follicles and make your hair stand up. It's a very, very minor thing in the skin, and in fact, for humans, has no real function. For animals that have fur, hair covering their entire body, these types of muscles are important. They help with temperature regulation. But humans just don't have hair covering their body, and so making hair stand up on end isn't actually that functionally important. It's been subsumed by other things in our body, like when you're anxious or scared or something like that, your hair will stand up, but it doesn't really do anything for your body. It just ended up being part of that response. Okay. More importantly, there are muscles in blood vessels. Now, blood vessels are organs of the cardiovascular system, and the cardiovascular system is invested in all of the different organs in our body. So we can talk about particular blood vessels being part of another organ system. So there's blood vessels that are very important for the skin, not just in supplying the skin with nutrients and clearing waste products away, but also in helping to regulate body temperature through the skin. And blood vessels, namely arteries, have smooth muscle in their walls. And that smooth muscle is very important in controlling how much blood is flowing through the skin at any given time. So there's a couple of places where muscle is found. The erector pili muscles that make hair stand up on end while they're there, and it is actually a component of the skin, realistically, it doesn't really do anything for humans just because humans don't have fur. And then the muscles that are in blood vessels, while it's important to how the skin works, blood vessels are kind of part of another organ system too, but they have to do with how the skin does its job, so it's there. And then finally, nervous tissue is very important for the skin because the skin is really a sensory organ. Okay? How we interact with the environment around us, our sense of touch, so to speak, is really based on the skin. And so there's nervous tissue there. Now, when we study nervous tissue in the nervous system, we'll talk about certain types of cells and all that sort of thing. All that we have in the skin are what are called nerve endings. So the actual cells are located elsewhere, but they're connected to the skin in what we call nerve endings. And some of those endings have specializations associated with them so that we can interpret very specific types of stimuli. Okay. So that's the nerve tissue that's there. Those are the tissues in the skin. We have representatives of all 4 different types of tissue here. And we're going to see that in every organ, in every system. But here's a good place to really spend some time thinking about it. The skin, like I said, isn't that exciting and is for the most part really just a membrane. There are 4 types of membrane that are mentioned in the tissue chapter, 3 of them that have epithelia. There's mucous membranes, serous membranes, and cutaneous membranes. And cutaneous membrane is simply the skin. It's a stratified squamous epithelium and its associated connective tissue. And that's really what the skin is. There are things incorporated into that to give the skin the ability to do a bunch of stuff. So we have other things. But at its most simple level, the skin really is just epithelium and connective tissue. Okay? And that's part of why I think of it also sort of, especially in terms of the practical, as being an extension of the tissues portion of the course. Now, what's important to make this bridge between tissues and organ systems is think about how these tissues inform our understanding of the function of the organ system. So I have here a list of the various functions of the skin. I probably need to go through and update this a little bit to make it align a little bit more with the textbook. But everything the textbook says is essentially here. Might not say it quite the same way, but these are the major functions of the skin. And we can explain how these functions work by considering what tissues are present in the skin. For example, the epidermis of the skin is a stratified squamous epithelium. And I'm going to ask questions about stratified squamous epithelium over and over again in this course, in A&P1 and A&P2. When I say stratified squamous epithelium, you should immediately think protection. Okay. So let's try this. I'm going to say stratified squamous epithelium, and you're going to say protection. Okay, ready? Call and response. Stratified squamous epithelium.
Protection.
Stratified squamous epithelium.
Protection.
Simple squamous epithelium. Thank you for not saying anything. Stratified squamous epithelium is primarily about protection. We find it in places where we need the most protection. And so obviously it's going to be part of the skin. If there were a more protective epithelium, that would be in your skin. But we do find stratified squamous epithelium elsewhere. There's 2 types. There's keratinized and non-keratinized. The keratinized is what's in the skin. Non-keratinized is found lining the esophagus and other parts of the upper digestive tract, and the wall of the vagina, and the opening to the anus, and those sorts of things. So it's protective in its own way. It's just a softer version of the tissue. As far as protection is concerned, there's a bit more than just the physical structure of that epithelium. It has some components to it that help. Let me zoom in on this. There's a physical, chemical, and immunological aspect to the skin. So the physical aspect of it is that it is a stratified squamous epithelium. You can't quite read underneath where it says that, but chemical barriers getting at the fact that there's some other things about the skin, the epidermis, that help to protect. One is excretions onto the surface of skin, whether it's from oil glands or sweat glands or something like that, have a slightly acidic pH, which helps to inhibit bacterial growth, which will help protect. And then also in the skin, there are cells that make a chemical called melanin, which helps protect against UV radiation. So best place to put that is under a chemical barrier. And then the skin's also an immunological barrier. Physically, it keeps bacteria from getting out, and then chemically, it inhibits bacterial growth. But then there's a special type of cell that when we get an infection, something's able to get through the skin, because you cut the skin or something like that. There's a special type of cell that will help to initiate the immune response and fight off infection that might happen after you cut yourself. So there's various things that have to do with it being protective. I mentioned a few other things here. I don't know that the book actually says blood reservoir exactly, but what that's getting at is that in several places in your body, and the skin's one of them, you have blood vessels that store extra blood. If you need blood somewhere else very quickly, then the reservoir of blood that we have in the skin, as well as a couple of other organs, is able to immediately get that blood into circulation. You don't have to make new blood. You have excess hanging around for when you do need it. A very important part of what the skin does is to help regulate body temperature. And there's two main components to that as far as the skin's concerned. The two bullet points underneath that, one says dermal circulation, which means the blood vessels in the dermis. When you're hot, more blood flows to your skin so you can radiate off extra heat. When you're cold, blood leaves your skin so that you can retain body heat in deeper layers as much as possible. The other thing there, the second bullet point says sweat. Just having blood in your skin to radiate off excess heat isn't going to cool you off very well. You also need to sweat, and you have sweat glands in your skin, and that's their whole purpose. The energy in your body temperature can be given off very easily by raising the temperature of sweat until it evaporates. So in cooling your skin off, an increased perfusion of blood to the skin and sweat have to go together. If one happens without the other, it doesn't do any good. Okay? If you sweat without the increased perfusion to the skin, you have cold sweats. Okay? And you might have experienced that when you've been sick or something like that, and it makes you feel colder than you want to be. Okay? If you have increased circulation without sweating, then you're going to get flush. Think about when you're embarrassed or something like that. Blood goes to your face, it changes the color of your face, but also makes your face kind of hot. Okay? So the two have to actually happen together to help cool you off. Cool you off when you're too hot. There's not an opposite of sweat for keeping you warm. You don't unsweat or anything like that. But dermal circulation does go both ways. Okay? When you're hot, blood goes into your skin. When you're cold, it stays out of your skin. Deep to the skin is a layer called the hypodermis, which is not technically a layer of the skin. It means below the skin. And it's made up of mostly fat tissue. And so when blood leaves the skin, it goes deep to that fat tissue, which helps to insulate the body temperature a little bit. So that kind of fits in with the body temperature regulation. The book that we're using doesn't really stress the idea that the hypodermis is technically not part of the skin. It includes it there. But it's important for how the skin functions.
so that's fine. Very minor point, I really should put this at the end of the list. When you sweat, you're filtering things out of your blood, so any waste products that are in your blood will get filtered out. We primarily do that in the kidneys, but anywhere that you filter blood, you're going to get waste products out. So when you're sweating, there are some waste products on your skin. It's a very, very minor version of what the kidneys do, but still the same idea. And I did mention earlier, the skin's important for sensation. And this is really the main part of what the nervous tissue in the skin does. It's where we receive sensory information. There is a little bit of nervous tissue that controls the smooth muscles in the skin, but primarily nervous tissue in the skin is about cutaneous sensation or sense of touch. And then finally, just to mention in the book, there's a nice little sidebar about this. Part of the metabolism of vitamin D requires the skin. Part of producing active vitamin D requires UV radiation for the chemistry to work. And the only place that we have high levels of UV radiation is in the outer layers of our skin. So this molecule that's going to be vitamin D goes through the skin to be modified before it becomes active in our body. Vitamin D is more important for bone health, but it's a function of the skin to produce it and make it help with bone health. And there's a sidebar in the book that describes that. So as far as the skin's concerned, those two things, the types of tissues that are found in it and how they relate to the functions, are the things that I think are most important. There are two other boxes in here I want to point out. One is just the physical structure of the skin. There are two main layers, the epidermis and the dermis. Like I said in the book, they do talk about the hypodermis and don't really do a good job of saying it's not technically part of the skin. I don't have a big problem with that, but just understand that these two layers are really the skin. The epidermis is the stratified squamous epithelium, and that stratified epithelium, its various layers, are segregated into different regions. And this picture here from the book highlights what the 5 different major layers of the skin are, and points out the different types of cells. I have a list here of those types of cells. Keratinocytes or keratinocytes, however you want to pronounce that, are the main type of skin cell, and they're found throughout all of the different layers. The other types are found only in specific layers. Melanocytes and Merkel cells are found at the base of the skin, right next to the dermis. And then dendritic cells, those are the immunological cells I referred to earlier. They're found in the layer just past the basal layer. And then keratinocytes are found everywhere from the base to the stratum corneum, the outer layer. The layers of skin cells, so we have the basal cells down at the bottom, and then this picture actually doesn't have the dendritic cell in it, although the book talks about them. I didn't draw one in the picture, but it would be found in this layer in here. And then here's a layer with all these dots. Notice beyond this layer with all the dots in it, the cells stop having very visible nuclei. Okay? And that's because these cells here are too far away from the blood supply. They're not getting nutrients, they're not getting rid of waste products, or they're not able to get rid of waste products. So they end up dying. And another aspect of the protective nature of skin, or the protective nature of stratified squamous epithelium, is that the apical-most cells are dead. They're physically too far away from the blood supply to survive, but they stay attached because they provide a layer of cells that you can't do anything worse to than already being dead. Okay? So when you scrape your skin or something like that, you're scraping those dead cells. Okay? If you scrape your skin really hard, it'll hurt because you might get through all the dead cells and actually start doing something with the living cells. But a minor scrape isn't a big problem because it's just going to affect those dead cells. But they provide a very protective layer so that the living cells and the rest of the epidermis are not exposed to chemicals and temperature and whatnot on the outside. In this picture, it doesn't look like a whole lot of tissue there, but really, I got to get back to that picture in a second. It can be a pretty significant portion. Oh, these are pictures out of the book, and now that I look at them, I'm not too crazy. The pictures from our slide set of the keratinized stratified squamous epithelium shows the stratum corneum is a much more significant layer. And in this one, these are two types of skin. So here, you can see the dead cells kind of flaked off up here, but they look kind of minor. The microscope slides that we have, that we captured last week, this component is a much bigger part of the tissue. And then here, there's not a whole lot. These two pictures of skin show two types of skin, what we call thick skin and thin skin. Thick skin has more tissue out here, which actually this picture is not a very good representation of still, but thick skin is found on the soles of your feet and the palms of your hands. Okay. Now, for four-legged animals, those are surfaces that they walk on, and you need thick skin when you're, on your soles of your feet or something like that. For bipedal upright humans, we still have thick skin on the palms of our hand because we still use those surfaces for grasping. It provides a great sort of friction-full, if that's even a real word, connection. If think about monkeys that are swinging around in trees, it's still doing the same thing with locomotion. And for us, grasping tools, whether it's a hammer or a smartphone or whatever, the thick skin provides a bit more friction with whatever we're grabbing onto, and helps add to that. So that's thick skin. Thin skin is mostly found everywhere else. The thinnest skin on our body is found in our scalp and right at the edge of the lips. If you've ever eaten very spicy food, you'll usually get a little burning sensation right at the edge of your lips. And that's because the skin there is so thin that the chemical that is spicy gets in, it can activate pain receptors there. So you can
So the lips and where else?
scalp. Yeah. So I'm not positive what this picture's of, but usually pictures of thin skin from textbooks are actually pictures of the scalp. Not that we can see any hair follicles there, but I need to go back to this picture right here. This is representing keratin. Now, keratin is a fibrous protein that's found inside these cells, and they're called keratinocytes because they produce a lot of keratin. These two pictures, there's a link down at the bottom, kind of in the very, very fine print, showing you where the picture actually came from. But these are two pictures representing the protein structure of two fibrous proteins. The bottom one is collagen. The top one, I used to always say is keratin, but I actually discovered when I was going through this stuff over the past week that it's technically not keratin. It's a related protein, but for whatever reason, they didn't have a picture of keratin to show you. But it does show you the basic structure. Fibrous proteins, whether it's collagen or something else, largely are defined by having protein strands that are wound around each other, braided around each other. The collagen has 3 layers. The keratin-like stuff at the top has 2, not layers, strands. But still, it helps to give them some strength. These two pictures are representing protein structure in the sense that each of the little spherical things there represents an atom. These two pictures are not to scale, which is to say they're both organic molecules. They both have mostly carbon in them. So one of the atoms here or one of the atoms here is most likely carbon. So you can see that the atoms on in the top one are smaller in representation than the bigger one because it's at a different scale. I thought I had a picture that I photoshopped to make them the same scale, but I couldn't find it. I must have not saved it when I upgraded my computer in my office. But still, the idea is collagen that we talk about in terms of connective tissue and keratin have some similarities just in their protein structure being fibrous. And that's what keratinocytes are named for, because they have a lot of this type of protein present. Now, keratin belongs to what we call, didn't want to go that way. Doesn't matter what's on the screen right now. Keratin belongs to what we call the cytoskeleton, and it's an intermediate filament. So the cytoskeleton in any cell has three types of filament proteins. There's microfilaments. All cells have microfilaments. It's a protein called actin. And there are microtubules. All cells have microtubules. But then cells vary in the type of protein that's called an intermediate filament. And keratinocytes have keratin as their intermediate filament. I'm trying to think. Neurons, the main cell in nervous tissue, have a dedicated intermediate filament, which I think is called, it's not this, but it's something as simplistic as neurofilament. Actually, that might be what it's called. Yeah, it must be called neurofilament. Anyways, so different types of cells have different intermediate filaments, and keratin is the one in these particular cells in the skin. What neurofilaments do is they connect all of the different attachment points within a cell to its neighboring cells, making a mesh inside the cell. So back in the tissue chapter, there is a picture showing desmosomes and those sorts of attachment points that hold epithelial cells together. The fibers that are attached to it that they don't really name in the picture of the tissue chapter, for the skin would be keratin. And it makes those cells very strong even when they're dead. And let me go to this picture actually after all. So up here, this flaky part of the skin is held together because all of these cells are interconnected through their filament, their keratin filament network within the cells. That's all they are. They're just cells full of keratin, which holds them all together. Okay. And it gives the skin some strength. So that's a little bit about the layers of skin. I spent a little bit more time than I thought I would there. But be sure that you're familiar with the layers of the epidermis. There are layers of the dermis too, but they're a little bit more simplistic. There's one up here that's rich in blood vessels, and then down here, which is the dense irregular connective tissue in the tissues that we looked at before. Also, this block here addresses things that are found, it says within the dermis, and actually there's a part of that that's not quite true. Another way of thinking about this, and maybe this would be a better subheading for this box, is extensions, no, not extensions, additional epithelial structures. We'll go with that one. So glands are epithelial tissue. Glandular tissue is that 9th type. And in the skin, there are actually 4 types of glands. Now in the book, they only deal with two types, sweat glands and oil glands, or sudoriferous and sebaceous glands. But they are associated with the skin. So I want to mention a couple of others, but they obviously are very specific glands, have very specific locations. Mammary glands produce milk. Ceruminous glands produce wax. Mammary glands are, of course, going to be associated with breast tissue, and ceruminous glands are found lining the ear canal. But they're still glands in the skin. And really, they're both variations on sweat glands. Sweat glands filter the blood to make sweat, getting water out of the blood and that sort of thing, and a few other things. Mammary glands do the same thing, but then add to that milk proteins and lactose, which is the milk sugar disaccharide. And then ceruminous glands do the same thing, but instead add to the basic filtrate from the blood, waxy secretions, which helps to trap debris in your ear canal, and then it moves out and helps keep your ear canal clean. There are two types of sweat glands. Now that I look at this, I realize that I have the names off a little bit. I think the textbook doesn't use the term merocrine, instead uses eccrine to describe one, and then apocrine is the other type. These are, there's two types of sweat glands, and in parentheses, hopefully you can see it says there, one is a regular sweat gland and the other is a smelly sweat gland. Apocrine sweat glands are found where we have hairy skin, armpits, groin, beard areas. And they produce sweat that contains additional material that leads to that being smelly sweat. It actually doesn't necessarily contain any odorous molecules in it, but rather what is contained in it is metabolized by bacteria on the skin, and they produce smells. I have this whole tangent to go into on that, but I'm not going to because we don't really have time. So those are different types of sweat glands, but really there are four major types of glands, and here in parentheses each one has its particular secretion listed there. I suppose there's a more formal name for what we call sweat, but we always just refer to it as sweat. Oil is also known as sebum, which is sort of the root of the word sebaceous. And milk, of course, we call milk. Wax is also known as cerumen, which is the root of the word ceruminous. But sweat, oil, milk, and wax are the major ways we usually refer to those secretions there. So those are glands. Now that I think about it, I'm not sure that I ever updated these pictures here. Nope, I didn't. So I think when I started having technical difficulties and I didn't put the narration in with this, was about when I was about getting to this picture. This picture is not out of our book, but it is showing different aspects of the sensory endings that are in the skin, just to show you some variety there. But there are various things, and they deal with different types of stimuli. Free nerve endings generally deal with pain and temperature sensations. A root hair plexus can sense when a hair moves. If you have an insect on your skin, you usually only know of it because when it's walking along your skin, it moves hair, and you can feel that through root hair plexuses. The Merkel cells that are in the basal layer of the skin, and this picture doesn't represent that actually correctly, but they associate with nerve endings, and together the Merkel cells and the tactile discs sense a particular aspect of touch on the epidermis. Corpuscles are referring to special endings wrapped around the nerve endings that help transduce particular sensations. So there are tactile corpuscles and lamellated corpuscles that transduce different types of pressure sensations in our skin. Lamellated corpuscles are really deep, and so if you really push on your skin for a while and you hold it there, and then when you let go, you can still kind of feel where that was. It's because of this structure here. It's kind of like an onion. It has a bunch of layers. It gets squeezed, and then it slowly goes back to its original shape. And as it's still compressed a little bit, you can feel that. So I still feel the spot on my where my thumb was pressed against my palm because of how these things work. Okay. The other type, I can't remember what it's called now. The other corpuscle I just went past really quickly, tactile. That's right. It has a very bland name for it. Deals with a different aspect of pressure. And then this corpuscle, I think it deals with stretch in the skin, if I remember correctly, but don't quote me on that. The way that it's drawn here suggests that it's kind of a stretch receptor. So, wasn't expecting to say this, but the different nerve endings, which are represented here, and these are just simple free nerve endings for the sake of this picture. They usually subserve, so this one nerve ending here branches out and connects to the skin sort of in this field right here, and another one here. They don't overlap. In some cases, they might overlap a little bit, but we can tell when something's touching our skin here or here specifically because different nerve endings sense that. If there are two spots, how do I want to say that? Let's say we take a pair of tweezers and you pinch the tweezers together so the two tips of the tweezers are very close and you push them against the skin, they might feel like one thing touching your skin. We can tell when two things are touching their skin when they're touching two separate receptive fields. So if we take some tweezers and we put them together and touch our skin and they're both touching the same receptive field, our brain only gets one signal. But it's when the tweezers are far enough apart that they're touching two separate fields, our brain gets two signals and we can tell that there's two things touching our skin. If you want to have some fun at home, you got some tweezers, see how far apart the tips of the tweezers have to be for you to be able to tell that there's two things versus one. And how sensitive your skin is kind of corresponds to that. The distance between the tips of the tweezer is going to be shorter on the finger pads and the back of your hand and your arm and your shoulder. Progressively from the fingertips to the back. You need more space between the two stimuli for you to even recognize that it's two things. And that sort of corresponds to how sensitive your skin is in different spots there. Like I said, the sensory ending picture there isn't out of our book, but I never got rid of it if you want to see some examples there. But then back to our book. The other thing is hair and nails, neither of which I'm ever that excited about talking about. And in fact, I'm not going to talk about them anymore, partly in the interest of time. You can go to this, the book and learn what's going on there, the structure of hair. Really, the hair is not unlike the epidermis. It's made up of keratinocytes. There are living ones, there are dead ones. There are melanocytes that release pigment to color the hair, that kind of thing. They just different structures, so it's not part of the epidermis, but it's related to that. Nails are kind of the same idea. Instead of the dead layers of cells that's on the surface of your skin elsewhere, the dead cells on your nails is a nail. So underneath the nail is the living cells. If you've ever gotten something underneath a fingernail and it's really painful, it's because it's getting into the living cells. But the dead cells on the outside where your nails are kind of the same as the dead cells that are on the outside of your skin. Hair and nails are different in that they have a different type of keratin in them. It's no longer referred to this, it's now referred with a number designation, but it used to be called keratin and hard keratin. Now it's keratin, I don't remember the numbers, but let's say keratin 4 and keratin 7 or something like that. And the difference between the two types of keratin is really how strong they are. So hair and nails have a stronger keratin in them, and so they don't fall apart. So the outer layer of your regular skin flakes off eventually, but you have to actually cut hair or nails to get rid of those cells. The hard keratin holds the cells together even better. Okay. When your hair's not growing very much, it's not because the cells are flaking off at the end, it's because the whole hair is coming out of the follicle. If the follicle remains intact and the hair keeps growing, the hair will just keep getting longer and longer. Okay. Same thing with nails. You can break a nail or something like that, but if you can protect a nail, it will grow longer and longer. You have to physically cut the ends of it off. When there are problems, they can be kind of brittle and they can break, but in a normal, healthy adult, hair and nails will just keep growing and they don't lose those dead cells the same way that the rest of the skin does. That's because it has a different type of keratin in it. So, now, that's mostly what's covered in the book. So the types of tissue, the functions of the skin, the structure of the layers of the skin, and the other parts of the skin like glands and nails and hair and that sort of thing. I have here something which the book doesn't address in that chapter, and these are not pictures out of the book, but and actually this particular book has a rather graphic set of pictures. So this poor kid's got a wound in his forehead here, and they're just depicting it here. Usually when we're talking about the skin, we also think about wound healing and how tissues heal. And what happens when there's a cut through the skin, there will be blood vessels in the dermis that are broken, and they'll bleed out, and that blood will fill the wound site and clot, forming the scab on the surface of the skin. As the scab resolves, the tissue will heal behind it. So in the third picture here, we have a scab there. It's only sort of half as deep as it used to be because the dermis has healed back behind it, and the epidermis, which is kind of the purple part, has grown around across the wound site. Now, epithelial cells will heal back almost to new. You can't really tell when an epithelium has been damaged because once it grows back, it looks exactly like it did before. And that's partly because epithelial tissues are always turning over. There's always cells coming off of epithelia and always new cells growing in. So when epithelia heals, it heals back good as new. Connective tissue doesn't heal quite the same way. Instead, where the cut was, fibroblasts migrate in there and make a lot of collagen to hold the wound site together. It's like the connective tissue feels really bad about having not done its job properly, and so it's growing back stronger than it was before. And all of the collagen that's in there makes up what we call the scar tissue. And so in the fourth picture where we have it healed, the epithelial layer actually isn't any different than it was before. essentially. But you can see the scar where the cut was because of the high density of collagen fibers underneath. The connective tissue is qualitatively different, and you can see that through the epidermis. The epidermis itself isn't different, but the dermis, because it grew back with more collagen, is also going to leave a visible scar that we can see through the epidermis there. So like I said, I don't think the book deals with wound healing quite the same way. There aren't pictures in the book. These are pictures that are different from a different book. But essentially what we can see when we cut through the skin, it's a good example of how different types of tissue heal. Epithelium heals back as good as new because epithelial always repair themselves. Connective tissues will grow back with a scar. Depending on how big of a wound it is, the scar might be visible. Okay, so a small cut, you won't see a scar probably because it's so small and eventually the collagen will resorb back as it was. But as long as it's a sizable cut and the scar tissue is pretty large, you'll always see it because it's it never has a chance to completely get back to its original shape. It's not mentioned here, but while I'm talking about wound healing, let me just mention muscle tissue doesn't heal back as well as epithelial tissue, but it has specialized cells that help it heal back. If you go to the gym and you work out, you're purposely damaging muscle tissue so that it will grow back. And when it grows back a little bit stronger. If you do something just heinous to a muscle and it's completely damaged, then you'll have bigger problems. But a muscle, more than just muscle tissue, will scar up. But if you're just talking about muscle tissue, it'll usually grow back and be a little bit stronger than it was before, which is the whole point of working out. Nervous tissue, however, doesn't heal so well. Nervous tissue in the brain and spinal cord does not heal. When we get to the nervous system, I'll talk about that a little bit more. But in the central nervous system, nervous tissue doesn't really heal. In the periphery, it can. So if there were a nerve going through this space here when it's cut, that nerve might be able to grow back, depending on exactly how it's damaged. But in the periphery, there are mechanisms to help nervous tissue heal. If you happen to look at this presentation, to go back and get some of this information, there is this little brief description of a case study here that just talks about a skin pigmentation problem. I'm not going to go through that. I don't use that anymore, but I haven't erased it from the presentation. So if you're interested in that story, you can look at it, but I don't address it at all in the assignment scale. So that kind of highlights the things that I want you to pay attention to with the skin. What I think is most important getting out of the skin chapter is more the idea of how different types of tissues lend themselves to the function of an organ system. Bless you. So pay attention to that. The basic anatomy of the tissue, the layers, the types of tissue, the cells that are found there, that's important, but you can get that out of the reading yourself. And then the specializations that are glands and hair follicles, that sort of thing, you can get more in-depth detail about that. So that's a quick rundown of the skin stuff. I had hoped, and I knew I wouldn't quite do it, to, in the first half today, then go on to talking about bones a little bit. Not going to be time for that. So we're going to take a break. And then when we come back, I'll introduce the skeletal system, and we'll look at what the skeleton's made up of, the specific bones. So we're taking a break a little bit earlier than we normally would, but we still want to take about a 20-minute break. So by that clock up there, be back here by about 10 o'clock. And then we'll get started up again with this bone stuff, so.